Well, hello and welcome to the first lecture in the course Next Generation Sequence Analysis here at NYU Poly. My name is Jonathan Flowers. I'll be your instructor for this semester, and if you haven't checked it out already, feel free to head over to the welcome page where I introduce myself in a little bit more detail. This introductory lecture is really just to provide some context for next generation sequence analysis, and also to familiarize you with some of the basic techniques, the machines that we use to generate the data, the scale of the data that we'll be talking about in this class, and I'll close with a beginning or an introduction to some of the vocabulary that we'll be using throughout the course. So as many of you know, in the last 5 to 7 years, the field of genomics has experienced a revolution in data acquisition, and this was really the product of some major advances in technology that led to the ability to massively increase the amount of DNA sequence we can collect with very little effort. So essentially, this next generation of sequencers is based upon a technology that consists of clonally amplifying small pieces of DNA or RNA, building what we call sequencing libraries, placing them on what we call a flow cell, and then using fluorescent chemistry and sophisticated imaging technology to build the sequence of literally millions of small fragments of DNA simultaneously. These, also called short read or massive parallel sequencers, also include technologies that we call single molecule real-time sequencing, led by a company, PacBio. I'll talk to you a little more about that in a second. And the entire field really is characterized by high throughput, the ability to sequence massive amounts of DNA from many samples rapidly and at a low cost, at least per base. As bioinformaticians, really, we are the expensive part of the sequencing process. The DNA per base now becomes quite cheap. The analysis downstream is quite involved and costly. So what do I really mean by high throughput? And I just thought I'd introduce you to a few numbers here to illustrate really what I mean by high throughput. So here I'm going to be illustrating this with the Illumina HiSeq. This is probably the main workhorse of the major sequencing centers, both at the Broad in Boston, in Cambridge, and at BGI in Beijing. Probably the two leading sequencing centers have these Illumina HiSeqs lining their factory floors, if you will. So the HiSeq consists of two flow cells. Really, these are small glass slides to which we attach the clone libraries. I'll be going into some detail about that later. There's 8 lanes per flow cell, and 350 million reads per lane is an obtainable number that we have obtained right at Washington Square Park and at New York University. We get about, we can sequence quite short reads with the Illumina HiSeq, so this is typically 50 to 100 base pairs per read. So if you multiply those 350 million reads per lane times the 100 base pairs, you end up with 35 gigabases per lane, or 35 billion base pairs. Continue to do the math, and one flow cell you can obtain somewhere around 280 gigabases fairly routinely. So the human genome is about 3 gigabases. So in one flow cell, if you take 280 and divide it by 3, that's close to 100. So that's 100-fold coverage of the human genome in a single flow cell. So the Illumina HiSeq is a workhorse because of the very cheap cost per base. However, there's many instruments out there that each of them have a unique contribution to the field of next generation sequencing. The PacBio, for example, offers longer reads, more than a kilobase in length. The Solid is a competitor of Illumina, offers a higher accuracy per base. And each of these have various aspects that are beneficial. So it's not that any one of these is necessarily the best machine, or it's really the machine that best suits your purposes. So another example would be the 454 here from Roche 454 Life Sciences. 454 is a technology that also sequences moderately long reads, around 400 base pairs, I think is typical. But it was the first longer read sequencing technology, and these guys back in maybe 7 or 8 years ago now approached Jim Watson, the co-discoverer of the structure of DNA. He lives out on Long Island. They approached him, and he was completely interested in having his own genome sequenced. And they demonstrated that you can assemble a genome very quickly and rapidly using their longer sequence technology. So frequently you combine technologies for a particular purpose. So 454, say, combined with the very high coverage you can get from Illumina, and this is a common strategy to sequencing, de novo sequencing genomes now. Long reads from 454, short reads from something like an Illumina HiSeq. I just thought I'd mention quickly, there are now benchtop systems. These are smaller units that are meant for speed. So in the next slide, I'll show you the times that it takes to run to do what we call sequencing runs on these machines. And these benchtop systems really are meant for doing exomes or at sort of high throughput, low coverage, but very quickly. Perhaps as quickly as build the libraries in the morning, sequence overnight, have your sequence the next day. So I thought I'd just walk you through just briefly to give you a sense of these machines, how much they cost, so they're not cheap. The Illumina HiSeq, you can see over here, $650,000. The amount of sequence you get per run, 600 gigabases. This is the number that Illumina throws out there, which wasn't far from the number that I quoted you a few minutes ago, which we routinely obtain somewhere around 550 or so gigabases. So here you can really see the cost per gigabase, $41. So the human genome is 3 gigabases, as I said. So now we can completely cover the human genome with an Illumina HiSeq run, okay, or excuse me, we can cover the genome for the cost of about $150 million. So this is per gigabase. The human genome is 3 gigabases, so 3 times about $50, $150 to get to 1x. But then we can, for maybe 100x, which is a quite deeply sequenced genome, we're talking somewhere around $10,000, which is a vast improvement over the multi-millions of dollars it took to obtain the very first human genome. Runtime here, you can see here there's some distinct differences. 10 to 11 days is very typical for an Illumina run. Here you can see the PacBio is quite a lot quicker. Just a quick look at the read lengths. Here you can see this single molecule technology of 1,500 bases, whereas the HiSeq, they're claiming 150. Still very typical to do 50 or 100 bases for each of these short reads on Illumina HiSeq. One final thing, I'll explain this a little bit more. Here, the insert sizes up to 700 bases for these technologies. More routinely, we use 300 to 500 bases insert sizes. So I'd just like to jump now to begin to tell you about some of the common terminology that we'll be using in the class. Some of these are quite obvious to many of you, and that's okay. This will be a good review. So what is a reference genome? So a reference genome is a representative genome of a species. It's typically generated via the old Sanger style or clone-based sequencing. The term BAC here is a bacterial artificial chromosome. This is a very large molecule of DNA, up to say 100 kilobases, that are placed in a vector. And the way that we get such large and complete reference genomes is to sequence just the very ends of this 100 kilobase or so size molecule. This then we can do that for many similar large molecules where we have the ends. By sequencing the ends, we know we have the linkage relationships of the sequences right on the end. And that's important because that's how you begin to assemble a genome, is to know the linkage relationships, particularly of distantly spaced sequences of DNA. So for the old clone-based sequencing, you'd sequence the ends of tens of thousands of bacterial artificial chromosomes that contain your genome or your DNA from your species of interest, and then you use a computer to assemble these fragments into oftentimes complete chromosomes, which is very different from how we do it today. So the genomes are typically haploid. That means that there's a single parental complement of the genome, say the copy that comes from either the mother or the father. And it's frequently the consensus genomes come from many individuals. They're high quality sequence, so the individual nucleotides, we have a lot of confidence in those base calls. And oftentimes they consist of contigs or scaffolds. So the distinction between a contig and a scaffold is a terminology you should know. Contigs are the smaller contiguous pieces of DNA, anywhere sometimes from as small as 100 to many thousands of base pairs. Scaffolds are pieces that we know have linkage relationships, but there are gaps between them. So a scaffold might consist of as few as 2 contigs with a gap in between them, or it could consist of many tens of contigs, all of which have gaps in between them, which together, the contigs with the gaps, make up the scaffold. So finally, the purpose of a reference genome often is what we call a backbone that facilitates the assembly of a closely related genome. So let me just give you an example of the human reference genome, GRCH37. This is the current build that people are working on. It's derived from 13 individuals from Buffalo, New York. It consists of 22 autosomes, sex chromosomes, X and Y, and the little known mitochondrial genome, which is only about 15 or 16 kilobases. It contains only about 250 gaps as of 2010. This is down from about 150,000 back in the original build in the early 2000s. So there's pieces that are not known where exactly they belong. We call those unlocalized or unplaced sequences. There's also alternative loci. So these are really copy number variants or pieces of DNA that are found in one individual that might not be found in another. So just since many of you will be working with these genomes over the course of your career, just a heads up that different research communities, while they're basing their analysis on the same build, they will modify that build to their own purposes. So the UCSC, University of California, Santa Cruz, they have the genome browser there. They have their own version of GRCH37, as does the 1000 Genome Project, which is the first really large-scale sequencing project of many human genomes. We'll be talking about the 1000 Genome Project throughout the class. and hopefully we'll even get to analyze a little bit of their data. So later this summer, we anticipate that Build 38 will finally be out. So how do we use a reference genome? Again, the most common approach is to use it as a backbone to which we can read map or align the short reads that we collect from our sequencing machines. It's frequently used in RNA-seq studies, so we sequence small RNA molecules with the purpose of quantifying the underlying RNA. And then what we do is we map the reads back to the genome so we know which gene each individual read comes from, and then we count the number of reads to estimate the copy number of the transcripts in the gene expression pool. Obviously, it's used in whole genome resequencing. So what we mean by resequencing is essentially the sequencing over and over again of multiple individuals of the same species. So this is the field of population genomics. And then, of course, the read can be used. The reference can be used to do what we call reference-assisted de novo assembly, where we're de novo assembling a species, but where we have a moderately close genome, but not closely enough related that you can use it as your typical backbone to do typical mapping to the reference. So there's many other applications. These are just some broad categories, and we'll come back probably in the next lecture where we'll talk a little bit more detail about some of the applications. So a common terminology we'll use throughout the course is paired end versus single end read libraries. So first of all, what I mean by a library? A library is the taking of a single sample of DNA and preparing it for sequencing. It's built into what we call a clone library, which is then placed on individual flow cells. We'll talk about that in a second. But single end reads is when, in the library construction process, we typically shear the DNA, we attach what we call adapters to either end of the sheared DNA. So here in red is an adapter, and the flanking sequence that we will end up sequencing on either end. And what we do is we, in single end sequencing, we attach both adapters, but we only sequence from one end of the molecule. In paired end sequencing, obviously, we sequence from both ends. So the blue part here is never really sequenced, or I guess in this cartoon, really we can say that the red, to be clear, is the adapter, and we sequence just barely into the blue from the red. But we don't sequence all the way across the blue, because the blue segment might be anywhere from 300 to 500 base pairs long, perhaps a little bit longer. But the point is that we typically only sequence very short reads, so between 50 and 100, or possibly as much as 200 base pairs. Occasionally you can sequence all the way across an individual insert, but that's usually pretty uncommon. So why do we use single end versus paired end? Well, single end really is just cheaper, and if all you need to do is identify a particular transcript, so each read counts towards the quantification of a single mRNA and how abundant it is. Single end can be very effective and cheap at quantifying gene expression. However, there's many applications where the paired end provides an advantage. One of them, as you remember about the BACs, is the strategy that if you know the two pieces of DNA are linked, then that really improves your ability to assemble genomes, although here these are very close compared to the BACs, and so not nearly as effective at BACs in generating large scaffolds or chromosome length sequences. But also another reason to use it even in RNA-seq studies is in alternative splice variant detection. So imagine if the two ends of a paired end library, one end is in one exon 1, say, of a gene, the second end is on exon 2, where that just tells you that exon 1 and exon 2 are both present in that particular transcript. So if you don't know about alternative splicing, it's very common in the human genome. And as an example, it's conceivable that for this example that exon 1 and exon 2 might be present in one version of the transcript for that gene, whereas exon 1 may be spliced to exon 3 in another transcript. So we can tell that exon 1 and exon 2 are actually part of this transcript when these paired end reads span the boundary between exon 1 and exon 2. In the other case, they might span the boundary between exon 1 and exon 3, which would tell us which alternative splice variant we're dealing with. So lots of other reasons to use these we'll talk about. Another one here is mate rescue. So it improves the mapping rate, or the proportion of reads that can be mapped to the genome. Because sometimes you'll get one read that doesn't map particularly well, either because there's been an insertion deletion event in the binding site of the read, compared to the reference, or maybe there's SNP variations, so mutations that cause one read not to map very well, but the other read maps, say, perfectly. So this allows us to do what we call mate rescue, where one read maps well, but this one sort of mapped, but that we have now confidence that this one maps to the right place because of just how well this other read mapped. So I just thought I'd introduce you to some terminology here. So the 2x100, this is a paired end run, the 2 meaning both ends of the read, and the 100 meaning 100 bases. And we, in a typical paired end run, can get 40 gigabases per lane from a total of 200 million reads. So I just thought also I'd give you just some information about, or just some terminology again. In this class, we'll refer to the two ends of a clone library as reads, whereas we jointly refer to both from the same clone as a fragment. Just a heads up is that many people use fragment and read, so the entire molecule, both ends, can be called a read in some circles, but I think it's much more common to call the individual ends a read. So, but we'll call together again a fragment. So 1x100 then, just to carry on the terminology here, is one single end read with 100 base pair read lengths in that sequencing strategy. A little more vocabulary, library indexing or barcoding. So this is the idea that we don't just typically sequence one library per lane. We can frequently multiplex, which means we can sequence multiple libraries per lane. So what we typically do is we take a sample, let's say it's an individual RNA-seq sample from a particular cell type, and what we can do is multiplex that sample by building one library for the RNA-seq or RNA library from one cell type. We can build it from a second cell type with a different library, but then we can combine those two libraries onto the same lane. So the way that we can distinguish the reads is by attaching the barcode to the adapter. So the barcode is a known sequence, and what we do is sequence through that barcode during the sequencing process, which allows us to then computationally infer which library the individual sequenced reads originally came from. So we do what we call computational demultiplexing after the fact. This is something that bioinformaticians will do. So I thought I'd give you very brief introduction to the library construction process. You can already see that I've inadvertently left out one piece of information here. I just thought I'd just alert you to the fact that there's a major problem in doing RNA-seq, and that is getting rid of ribosomal RNA, which is the type of RNA that is the most abundant in the cell. Up to 90% of the cell is made up of ribosomal RNA, which is not the type of RNA that biologists are most interested in. So biologists are more interested in the 20 to 25,000 protein coding genes, which produce mRNA, which then again goes on to be translated into protein, not the ribosomal RNA, which, there's only two primary ribosomal RNAs in the nuclear genome, and they're just found at very high copy number, they're repetitive, and they make a lot of RNA. So one of the challenges in getting good RNA-seq data is to remove the ribosomal RNA from the sample prior to sequencing, so that you don't waste all of your sequencing dollars sequencing ribosomal RNA. This is done oftentimes by poly-A selection. What we do, the part that I skipped, is to convert the RNA into complementary DNA, so we don't actually sequence the RNA. So the word RNA-seq is a little bit misleading. What we do is we first biochemically convert it to DNA. However, because they complementary base pair each other, we can infer back what the RNA was from the DNA sequence, but we end up sequencing the DNA. So I just probably should have put the cDNA step here. We first shear the RNA, then we make the cDNA. We, again, ligate adapters like we've talked about. We do a PCR amplification step for technical reasons to improve the density of clusters on the flow cell. I'll tell you about that'll make more sense in a second. And then finally, we have to quantify the libraries in order to optimize, again, the formation of clusters on the flow cell. So many of these steps are overlapping between the RNA-seq process and the DNA-seq process in the lab. So a couple more slides here. I thought I'd give you some insight into how the sequencing process works. And I'll have to say that Illumina is not particularly transparent in the extreme details of the sequencing process, because this is a proprietary chemistry of theirs. But we do have a general sense of how it works from reading the publications that they put out and the reading, just from going to their website. So again, in a typical sequencing experiment, we first digest a genomic DNA sample into many smaller bits, again, maybe 200 to 500 or occasionally a larger pieces. We then ligate the adapters, represented here by the pink and the purple adapters. You can see here at this point that the DNA is double-stranded. This here is a representation of a flow cell, of which a segment of it we could consider to be a lane. And the key thing to understand here is that this flow cell is incubated with small DNA primers that are actually stamped onto the flow cell, sort of like a micro array, if you're familiar with that technology. But it's a small single-stranded piece of DNA that is physically bound via chemical bond to the surface of the flow cell. That's a single-stranded molecule. So what we then do is to hybridize the original DNA samples here. And this is a little bit misleading because the pink would say hybridize with another pink one of these primers, and that primer provides a 3' end to which can be extended by DNA polymerase, which will build the A's, T's, G's, and C's along this template strand. So we have an elongating a molecule here during the course of the sequencing process. So imagine the primer here, free 3' end. What happens is that the flow cell is, at the beginning of the run, is incubated with A's, T's, G's, and C's, and each of which has a different color label. And they will bind to that free 3' end and complementary base pair with the template molecule. So what happens then is that each of these different clusters is binding, or each of these different little primers is binding a different piece of DNA. And what is happening effectively is that during the course of the run, the DNA is first amplified on the flow cell, and then sorry, I sort of glossed over the amplification step. It looks like there's one molecule here, but what's happened is the PCR works right on the flow cell to increase the density of a single molecule around in what we call a cluster. And then we begin the sequencing by synthesis, where the individual nucleotides are built. So in the first cycle, we image the entire flow cell after the DNA has been incorporated, and each of the spots or the clusters will be a different color. Okay, and this is how we infer the sequence. So a color, maybe one spot's a green. Green was bound to, say, the nucleotide adenine, and so we know at that position there's a green, or an adenine at the first position. So then we remove any unbound nucleotide. We then essentially wash the flow cell again during the second cycle. Again, the complementary base pairing between the nascent strand and the template strand of the corresponding complementary nucleotide. And again, a imager comes in, photographs the surface of the flow cell, and again we have different colored spots, again representing the nucleotides as they're being incorporated into these individual clusters. So that's a lot to say here. This part here is really how very slick chemistry, which we don't have a lot of detail about, because it's Illumina's prized chemistry for how do you do the second strand sequencing in a paired end run. You have to this is what's called bridge PCR. The molecule actually flips over, attaches to an adjacent primer or set of primers that are bound here. Again, we go through an amplification step to make this new cluster here, but now we use the 3' end of this new primer here to sequence the opposite, going the opposite direction here on this on our template DNA. Again, it's the same process. You incubate with the nucleotide as that are dyed. Imager comes, sequences the surface of the flow cell, and infers the color for each of the clusters. So over the course of many days then, we end up getting many images, very high resolution, and a lot of different colored spots, which then computers can infer the nucleotide sequences. So that's kind of hard and fast. There's be plenty of time to talk about that more in detail, or to do a little bit of reading on your own. It certainly will have opportunity for questions when we meet at our weekly webinar. So I think that'll be it for this initial lecture. Again, if you haven't seen the welcome video, head on over there where I introduce myself in a little bit more detail. Thanks a lot for listening, and I'll see you in the next lecture, where we'll talk a little bit more about some of the applications of next generation sequencing.